Hello students, welcome back to my channel CSIR Buddy, and today we are with another video. It is the continuation of the last video. In the last video we studied about autonomous and conditional specification, and now we are going to continue this topic with some experiments related to the same. Experiments related to autonomous and conditional specification. So this is a quick recap. Autonomous specification, cell which do not change their fate according to their surrounding. तो हमने पिछले वीडियो में देखा था कि when a cell has already got its cytoplasmic determinants, so wherever you put that cell, in whatever situation, it won't change its fate. Like if this is a muscle cell and if we put it into a liver cell, an environment which is responsible for making liver cell, then also it won't change its fate and become a muscle cell. So this is known as autonomous specification. And then we studied about conditional specification. Cell is not predetermined, so there is no predetermination. So conditional means in a certain condition it may change its fate due to some external condition or the condition of the surrounding. It may change its prospective fate. So if we bring a brain cell and we extract a brain cell and keep it into a environment of a liver cell, then the brain cell will change its fate and become a liver cell, will be converted into a liver cell. So this was about conditional specification, and we have already seen about the autonomous specification. Now Weisman and Rowe experiment, they did an experiment for the conclusion to make what is autonomous specification. So they took a egg. So the picture is not clear, so I will write it down. They took a fertilized frog egg. So they took a frog egg, and if it is fertilized, then it is known as a zygote. So the zygote got cleaved. So this is cleavage. Got cleaved, and there were two halves. It was divided into two halves. So this stage is known as two-cell stage. Now what he did is he took a hot needle and injected this needle into one half of the cell, and the other half was having living tissues. So one half got dead tissues, so this is dead tissue, and another half was carrying living tissue. So it was seen that this one half got destroyed half or dead tissue. So in the blastula stage, when cells were dividing, so what happened is the half which was injected with a hot needle died after division, but the part which wasn't injected or was having living tissues got specialized and got converted into Nerula stage. So we can see that this one particular part had no influence in this one half. So what we can conclude from this is that this half may have got its cytoplasmic predeterminants. Therefore it won't get conditioned, or it won't get differentiated due to certain condition. So this half has no influence over the another half which got destroyed due to injecting the hot needle. So this was the experiment to show the autonomous specification. I think this experiment is clear to you all. Then moving further, now Drich experiment. He experimented in order to find the conditional specification, to show the conditional specification. So he also took a cell, he took a sea urchin in its 2-celled stage, and then he separated these cells. And what he saw, that both the cells converted into a whole larva, which is known as pluteus larva in the case of sea urchin. So it got converted into pluteus larva. So he concluded that the fate of a nucleus depends solely on its location, कि किस लोकेशन में उसको रखा गया है. So sea urchin harmonious equipotential system. It is showing a harmonious equipotential system. Here the environmental condition or the condition in which the cells are put it on decides their fate. So if we consider a 4-cell stage, this is a 4-cell stage, then what will happen? These cells will interact with each other. These cells are interacting with each other, and when the cell interact with each other and then decide their fate, that is known as conditional specification. Therefore, when they were separated, they didn't have any predetermined or any cytoplasmic determinants, as we saw in the case of autonomous specification. Herein there was a predetermined cytoplasmic predeterminants which was determining their fate, but in this case they are solely dependent on the location where they are present. So maybe this cell's location is responsible to make a muscle cell, and this one is liver cell, suppose liver cell. This may be a heart cell, and so on. This may be a nerve cell, and these cells which were separated, they may have got divided further. They got divided or cleaved out, and then they changed themselves into a whole pluteus larva. So this shows that there wasn't any predetermination, any cytoplasmic predetermination. Therefore, even if we separate these cells from each other, then also each and every cell is capable of making a whole pluteus larvae. So this was an example, this was an experiment done for checking the conditional specification, for showing the conditional specification. So I hope this video was clear. Thanks for watching. If you like this channel, please subscribe to our channel, share this video and press the like button. Thank you so much.